Hello, and welcome to another episode of the JASN podcast series, a monthly podcast which we highlight an exciting paper from the current issue of the journal. I'm Alan Yu, Professor of Medicine at the University of Kansas Medical Center and Deputy Editor at JASN for Basic Science. Today, I am joined by Dr. Tobias Hermle and Dominik Spitz to discuss their interesting paper entitled A Drug Screening Platform for Podocytopathies and Drosophila Nephrocytes, which appears in the current issue. So to start off, can you please introduce yourselves and tell us a little bit about your background?
Hello, everyone. First of all, thank you very much for having us here today. It's really exciting to be part of that podcast. My name is Tobias Hermle. I'm a clinician scientist. I attended medical school in Tübingen, Germany, underwent clinical training in nephrology in Erlangen and Freiburg, Germany. And I also did a good deal of basic research. I did a postdoc here in Freiburg in a lab studying Drosophila, and also at Boston Children's Hospital studying genetic podocytopathies. At the end of 2017, I returned to Germany, starting my own lab, becoming an attending at the University Medical Center in Freiburg, and also becoming part of the faculty at the University of Freiburg. And my lab studies genetic podocytopathies with several models, but the Drosophila model takes the center stage.
Yeah, and my name is Dominik Spitz. I'm a biologist and a PhD student in Toby's lab for, now 4.5 years. And at the moment, I'm slowly but steadily approaching the end of my thesis.
Terrific, thank you. So let's jump into the science. So podocytopathies are proteinuric disorders driven by injury to the glomerular podocytes, like minimal change disease and FSGS. And there's been tremendous progress in understanding the genetics of inherited podocytopathies, which has led to progress in elucidating the cell biology. But most of the current drugs we use to treat these, even today, are non-specific immunosuppressants. So in this paper, you advocate for a drug screening approach. So can you talk us through the merits of drug screening for discovery, and what are the limitations of current technologies when applied to podocyte screening?
So, as you know, CKD is a major global health challenge. It affects about more than 8% of the global population, and its etiology is very diverse. Quite often, we don't really know what is the cause of the disease in our patients, and we cannot offer them proper targeted therapies. As you outlined, there is a good deal of immunosuppressive therapy. There are ways to manipulate the glomerular hemodynamics, but we have very little ways of really finding out how we can make the patient in a specific situation better. So there is a therapeutic gap. We're lagging behind compared to other fields such as cardiology or in cancer field. And one of the reasons is that the conventional drug screening approach is not very well designed for podocyte diseases. The conventional approach uses a very stripped down assay to look at a target. So you have a very simplified model, and then you have a target where you're sure of that it really affects the disease. And then you can screen millions of compounds to find out how that is better. But for the podocytopathies, we have a paucity of known drug targets. Quite often, we don't understand the disease well enough to have a good target. And then it's very, very difficult to model something as complex as a podocyte with its signature feature, the slit diaphragm, in any in vitro setting. And so that is one of the reasons why we're falling short on offering options here. And there is not, to my knowledge, a single drug that really has a podocyte-specific target at this point. So I think we need a better way for having a drug discovery.
Thanks, Tobias. So you kind of alluded to here the distinction between target-based screening and phenotype-based screening. Do you want to elaborate more specifically on that difference so the audience understands that clearly?
So in the phenotype-based screening, we bring an entirely different model that we use for screening. Here we, in our case, really a whole animal. So we have a much more complicated biological system that we're looking at. And that allows us then to really mimic a phenotype directly. And in such a system, we have the entire complexity of life. We have different cell types, different organs that really communicate with each other. And in particular, we don't need to know the target. Like, we can find a way to improve the phenotype and then figure out which is the target, which way that was made better later on. And that is very much more useful to looking at podocyte diseases. Inherently, the throughput is much, much slower. Like, you cannot study millions of compounds in such a setting, but the quality of hits is higher because it was already then validated inside a proper biological system that is an animal.
Terrific. I think you kind of answered my next question, which is what are the pros and cons of this? So it sounds like the cons are the throughput and the pros are that you can model the complexity of the system. So did you consider other systems, or you jump straight to Drosophila because that's what your lab works on already?
Yeah, I personally it was a serendipity that I stumbled upon Drosophila. I just joined a lab that studied that, and it sounded like a fun thing to do to me. But in my previous postdoc, I was looking at planar cell polarity, and that was really far away from any clinical work. And so I was very excited about the seminal Nature paper in 2009 by Denholm and colleagues that showed the nephrocyte as a podocyte model. And so I really wanted to work with that. And then I established that model as a model for the genetic podocytopathies in Friedhelm Hildebrand's lab. And then coming back to Germany, that was my first plan that I really wanted to look at. It was the first grant I ever wrote. And it was a few years in the making now, but I think it came together nicely. Now, why would you want to use Drosophila for such a model? It's a very simple stripped down model. It's small enough to really fit in a dish, so you can put it in a 96 well plate and feed the drug by having the flies in liquid food. It has a great genetic toolkit, so it's easy to manipulate pathways, and these pathways are simpler because you have less paralogs. And it has a very, very high turnover. Like, you can produce many of these animals. It's not restricted by any regulations. And that makes it a good model organism that has a good conservation as well of the genes you want to look at. About 70% of the disease genes are conserved. And then, of course, you have the nephrocyte. The nephrocyte is, in a way, a podocyte-like cell in that it removes toxins. I mean, this is why we have glomerular filtration, right? We want to remove any toxins, and we do that by filtration. And the nephrocyte does something similar. It works through endocytosis. And before endocytosis, you have a filtration barrier that keeps the large plasma proteins of the fly out from membrane invaginations where the endocytosis is happening. And the important thing really is that this model has a molecular similarity to the slit diaphragm, really down to the structure of these molecules in a multi-protein complex. You have a molecularly conserved slit diaphragm. And so altogether, this was why we figured, yeah, that's a very attractive system that can be applied to what we want to do here.
So to interrogate the function of the filtration apparatus, you developed flies transgenic for a fluorescent protein. Can you walk through how this worked and why you think it worked as a marker of filtration function?
So, as Toby already mentioned, the way the nephrocyte functions is that it takes up molecules for degradation from the surrounding hemolymph. And this constant endocytosis is the nephrocyte's main job, kind of. If we now want to investigate how well the nephrocyte actually works, we look at the endocytosis of a tracer as observable and functional readout. So typically we do this by dissecting the nephrocytes and presenting them with such a tracer ex vivo. And this is not really tricky to do, but in the context of a drug screen, of course, it's just way too time consuming to do it like that. So therefore, we generated this transgenic fly that ubiquitously expresses a GFP throughout the whole body, in our case, the larva. And this GFP is fused to a signal peptide that allows secretion from the cells where it is produced into the hemolymph. And from there, the GFP can then be taken up by the nephrocytes after filtration through the slit diaphragm. So for the screen, we just had to dissect the nephrocytes from these transgenic larvae, fix them immediately without the necessary steps for an ex vivo tracer presentation. And then we could just evaluate the intracellular GFP signal as a quantitative marker for nephrocyte function.
Terrific. That's very clever. Now, obviously, one can only show that the drug is working if it's applied to a diseased or dysfunctional nephrocyte. Right. So the other innovation here, as I understand it, is that you use a background of a RAC1 gain of function mutant. So can you explain why you chose this? And in this context, how generalizable do you think drug would be the drugs identified with this screen to other podocytopathies?
Well, exactly as you said, to see an improvement by a compound, of course, you first need to disturb nephrocyte function. So as one of the master regulators of the actin cytoskeleton, a tightly controlled RAC1 activity is very important for the complex architecture of a podocyte's foot process. And now there are several hereditary podocytopathies that are linked to RAC1 overactivity as cause of podocyte injury, and also RAC1 overexpression in podocytes in the mouse model recapitulates such podocytopathy phenotypes. So therefore, we checked if RAC1 overexpression gives us a corresponding phenotype in nephrocytes as well, and indeed, tracer endocytosis was reduced, and also the slit diaphragm distances on the nephrocyte surface were increased. So a phenotype that corresponds to foot process effacement in podocytes. And as at the same time, RAC1 gain of function did not just completely kill the nephrocytes, we chose this genetic background for our pilot screen. And on your question on how generalizable identified drugs are, in our case, with our screen hit against the RAC1 overexpression background, there might be some benefits for podocytopathies that have a disorganized actin cytoskeleton as the underlying reason for the specific disease. But with our whole animal screen, we test the overall effect of a compound on the biology of a whole animal. So this cytoskeleton does not only play a role in genetic podocytopathies, but in a number of contexts in podocyte biology. And within the podocyte, dysregulation can be faced in a wide range of scenarios. So therefore, a hit in such a background may very well have effects on other related situations too. And because you may identify a compound that stabilizes the system as a whole rather than displaying a very specific effect in this one specific factor, you may have some translational utility there. And of course, the main idea of using this Drosophila-based screening platform is that you can use all those different available genetic tools that Toby talked about that are out there for Drosophila research and just combine them with this model to generate any specific background you want to study.
Thanks. Yeah. So basically, this would be a sort of common downstream pathway that you'd be hitting. Terrific. Yeah. No, that sounds very powerful. So after you established a method, you did a pilot screen, and the first thing that happened was you had a positive hit, Zachopride, I believe it's pronounced, and you did a series of studies that validate this finding. And I know it's a pilot study, but I'm sure the audience are curious to know what's Zachopride. Can you say much about how it works, and is it even a promising drug candidate?
Well, yes and no. So Zachopride emerged as a hit candidate from our pilot screen, and it improved both the reduced tracer uptake and, very importantly, the disturbed slit diaphragm architecture as well. Now, Zachopride is an interesting molecule with a quite complex pharmacological profile. It has reported antiemetic, nootropic, and anxiolytic effects in animal models, And apparently back in the 90s, it was rather unsuccessfully clinically tested to treat schizophrenia symptoms based on its effects on serotonin receptors, both acting as agonist and antagonist on those receptors. And it also has agonist effects on inward rectifying potassium channels. So therefore, we checked these molecular effects genetically and pharmacologically on our model, but we couldn't find anything related to the reported effects. We also found that saccharide did not directly inhibited the REC1 activity, suggesting that REC1 is not its direct target, and that the mode of action of saccharide is probably a pleiotropic one with a yet unknown molecular target and maybe even a systemic effect. So how saccharide is able to apparently attenuate excessive REC1 activity and to improve the slit diaphragm architecture, we do not know at this point, but it may become an interesting substance for podocytes with potential translational utility for podocytopathies displaying cytoskeletal disorganization. Of course, given that additional testing of saccharide in, for example, the mouse model confirms our observations. And really, to our pleasant surprise, this hit could not better demonstrate the benefit of such a phenotype-based screening system. We do not know how saccharide improved our phenotype. The efficacy of this drug in restoring the slit diaphragm architecture and nephrocyte function is absolutely not anticipated at all by its pharmacological profile. And apparently the effect is not via its known molecular targets, but yet it works. And we see an improvement, making saccharide a valuable candidate.
Terrific. Well, sounds like more work to be done for the next grad student in the lab. So finally, now you've developed this method, how do you plan to move forward as a lab, and what are the next steps for you and for others in the field towards discovering new drugs for podocytopathies?
We're happy that we have this platform established, but I think the next steps will really require a more collaborative effort. We really would need industry partners to really move to an industrial scale with such screening. There's many steps that could be really improved on, like, for example, the sorting of flies of the right size, of distributing them. The imaging could be automated. The quantification could happen entirely automatically. So we could really scale this up to make this really a setup that you can actually use for drug discovery. And so, yes, we would be very happy if this would be the start of something that could really make a difference, and then really be using this fly model in different genetic backgrounds for a very wide array of podocyte diseases.
Terrific. Well, Dr. Hermle and Dominik, just want to thank you so much for helping us to understand this exciting new technology you've developed. Congratulations on a very elegant paper. Good luck, Dominik, on graduating soon. And thank you so much for joining this podcast.
Thank you very much.
Thanks for having us.
Today, Ross.
This podcast is copyrighted by the American Society of Nephrology. All rights reserved. All content in this podcast is for informational purposes only and is not intended to be medical advice. This podcast should not be used in a medical emergency or for the diagnosis or treatment of any medical condition. Please consult your doctor or other qualified healthcare professional if you have any questions about any medical condition or before taking any drug, changing your diet, or commencing or discontinuing any course of treatment. Thank you for listening to this podcast from the American Society of Nephrology.